Yes, so in the last lecture, we studied about the Mendelism and the principles of the genetics as well as variation. So in this chapter, we are going to study about that what is that material which is responsible for the inheritance. In the whole lecture of the principles of inheritance and variation, we studied about the different way how the inheritance is going on, how the sex determination is being done, how the X-linked chromosomes are giving disease in the next to next generation. So, we studied about all this. Okay. Yeah. But still, there was a one question, and that was what is the molecular basis of genetic material? So, which molecule is acting as a genetic material? That is still unsolved. And that's why this chapter is about it. So, in the Mendel's chapter and Mendelism, what we learn is that, let me open my whiteboard. In that, we have studied that there was something which is named as a factors. Then after it was known as another thing which is known as allele. Okay, yeah. So in that chapter I told you that what is written, this one is known as what you can read this. It's a 101, it is 101, it is 101, it is 0 in the module. It may be read as a lol. Yes, okay. But yeah, this will remain same to same. In such a way, whatever the material responsible for the inheritance, that material which is responsible for the inheritance remains same to same. The material for the inheritance will remain same to same. Okay, but at the different times, it was given a different names. Say for example, some said it is known as a factor, some says that it is not a factor but it is known as a allele. Yes, some of the scientists say that it is not allele, it is known as a trait or a gene. Gene is what segment of DNA. So as the time go on, the new discoveries came in the existence. Okay, and segment of DNA, gene is equal to, and they are present on what? They are present on what? They are present on chromosomes. They are present on what? They are present on chromosomes. So this is what we have done. Yes or no? Yes. Can you recall this? Yes. Huh. Okay. So here the question is that why we say that it was DNA only and was nothing else. So that's what we are going to study over here. That why it is a DNA only. Okay, yeah. So in some of the organisms it is a RNA also. Some of the organism it is a DNA also. But yeah, most of the organisms are having it as a DNA. Okay, yeah. So let's study what are the structure of the DNA, how we can study about the DNA. What is DNA? I already told you it is a type of a nucleic acid. Why it is given a, such a name nucleic acid? Because it is found in the nucleus. As it is found in the nucleus of the cell, that's why such acids are known as nucleic acids. And in 11th grade we have already studied about it, and we have studied about the nucleic acids. They are made up of a polynucleotide chain, especially in the chapter of biomolecules. Yes or no? In biomolecules we know about the DNA structure, as well as in your lower standard like 10th grade also you know about the DNA structure. Okay, but here I am going to give you a final full detailed structure of DNA. Okay, and that is what this chapter, and that's why the name of the chapter is what? molecular basis of inheritance. Okay, so let me share with you my presentation. So PPTs. Okay, I would just like to open my PPT. So this is what the chapter name is: a molecular base basis of inheritance. So here we are going to start about the molecular basis of inheritance. Okay, so earlier the people were confused that it might be happen that DNA is responsible for the inheritance. It might be happen that the RNA is responsible for the inheritance, and there were some scientists who were believing that protein is responsible for the inheritance. Okay, so it was a really a great confusion, because in the case of Mendel, in the era of Mendel, nobody knows, and the microscope was not there. Microscope was not there, so there was no proof about the molecular basis of inheritance, and that's why it's very important. Okay, yeah, so in this chapter we are going to study about 3 4 main things. Okay, I will tell you like what kind of topics we are going to study in this chapter. Okay, so here, first thing is that we will study about the structure of the polynucleotide chain. Then after DNA, then after search of genetic material, how do we get to know that the genetic material is this one only? There are some organisms in which RNA is acting as a genetic material. That is known as the RNA world. Then after replication. Hey, can you tell me what do you mean by this replication? Anybody know? I already told you earlier, huh? Even in the Mendelism chapter also I told you about this. Okay, can you tell me what you mean by this replication? Anyone? Anand, Hirali, Karina, Shreya. Anyone can tell me what you mean by replication? I told you to write down the whole page about this. Okay, what do you mean by the replication? What do you mean by transcription? What do you mean by translation? I have told you to write down the whole page about this. Replication meaning is what? When DNA is giving rise to DNA, that is known as a replication. Okay, so just keep this in your mind. When I told you about mutation and all those things, I told you about this, that if the gene, that is the segment of DNA, is undergone some mutation. Mutation means what? Any permanent change takes place in the sequence of DNA. Okay, yeah, then it is going to be happen. So when DNA give rise to another DNA, that is known as a replication. Okay, then transcription is known as a DNA to RNA. It is known as what? DNA to RNA. So when DNA give rise to RNA, it is known as a DNA give rise to RNA. It is known as what? Transcription. Is there, do you know any other organism who can do reverse of this? HIV, very good. HIV can go for reverse transcription. Okay, yes. So DNA to RNA is known as what? It is known as a transcription. Okay, DNA to RNA is known as what? Transcription. Okay, yep. Then translation means what? Different kind of RNAs will give rise to different kind of RNAs will give rise to protein. So different kind of RNA give rise to protein. So if the RNAs give rise to a protein, then it is known as a translation. That protein can act as either enzyme, or it can also act as a peptide hormone. Either it can act as a enzyme or it can act as a peptide hormone. Okay, So this is the protein is being synthesized, which is taking place on the ribosome, which is taking place on ribosome. So again, replication means DNA to DNA, transcription means DNA to RNA, and translation means RNA to protein. And that protein can act as a enzyme as well as peptide hormones. Okay. Then after we study about the human gene expression, regulation of human gene expression, human genome project, and DNA fingerprinting. So yes, it is one of the lengthiest chapter of the whole syllabus, as well as it is one of the conceptual ones. So I will be little bit slow, but you have to commit me that you will be always remain with me and you will not keep anything in the pending. Are you promising me? Honey? Because this is most IMP chapter. Okay, there are lots of the people who wants to make their career in the genetic engineering, genetics, biotechnology, and whatever kind of a research is going to be takes place now, it will affect this particular topics. Okay, so be very very strong in this topic. That is making your fundamentals very good, and definitely you will remembering me very positively because of such a chapters conceptual clarity coaching. Okay, yeah, okay, so let's start and let's go further. Okay, so the next is about what? So we are going to study about one thing, and that is DNA. In detail we are going to study about the DNA. Okay, yes, so here you can see this is the DNA. Why the DNA and RNA are known as a nucleic acid? They are known as a nucleic acid because they are discovered from the nucleus. So nucleic acids, that is DNA and RNA, are the building blocks of genetic material. DNA is a genetic material in most of the organism. RNA is also genetic material in some of the viruses. But those kind of viruses are known as a retrovirus. Such a virus are known as what? Retrovirus. What do you mean by retro? Retro means old. So that very old viruses are known as what? That's known as a retrovirus. And one of the example of such a retrovirus is corona. Corona is also a retrovirus. Okay. Okay. So RNA is mostly function as a messengers. In what it is acting as a messenger? In the process of translation, it is acting as a messenger. So it is just give message so that a particular amino acid will come combine with each other. पर सर कुछ पता नहीं चला. अरे डोंट वरी यार, I will show you, I will explain you very nicely because we have whole topic about the translation. Okay, so don't worry about that. But at that time the RNA will act as a messenger. Have you seen the messengers? Let me give you best example of the messenger in our Gujarati thali. So, if you are going out to eat up the Gujarati thali in a Gujarati dining hall, then what will happen? So, there are some manager, and manager is handling all the waiters. Okay. So, if you ask, if the waiters are keep coming and you are getting your food items at time, that's fine. Okay. But suppose you want something else and that's not coming in the rotation, then you will just move your head here and there. If you are looking for someone, then that manager will come. Yes, sir. And you have to say, I want chapati, okay, roti. So immediately that person will give the message like this. So this is the sign for chapati on table number 3. So that's how the manager as a messenger RNA, that is known as a mRNA, is conveying message to the tRNA. tRNA is known as a transfer RNA, which can be resembled with the waiter. Yes. Okay, and that's where one by one different amino acids will come. Amino acids are the building blocks, or you can say monomers of polymer whose name is. So many amino acids will give rise to what? Yes, again, protein. Yes. So if the protein is a polymer, then monomer is what? Amino acid. Honey. Okay. So let's go further. And what we have here, structure of polynucleotide chain. So, I would like to show you something better than that of this. And that's what I had explained you earlier also. Okay. So this is a good one. Yeah, the polynucleotide chains are the polymers of nucleotides. DNA and RNA are polynucleotides. Nucleotide have a three component: nitrogen base, pentose sugar, ribose in RNA, deoxyribose in DNA, and phosphate group. But it is better to show you the whole sequence. Okay, and let me show you the whole sequence, and for that I have to share with you my board work. Hey, are you ready? Yeah, and it's a very much fundamental thing, and you will love it. Okay, that's my guarantee. Okay, yes. So here you can see that. Yes, can you see? This is the fundamental one. So what is it? So you can see that here there is a nucleic acid. Okay. Yes, nucleic acid. Nucleic acid is known as a deoxyribonucleic acid, that is known as a DNA. Ribonucleic acid, that is known as a RNA. And please be with me. I will give you time to copy this. Okay. So just be with me. Okay. Just enjoy this. Don't write down anything right now. Yeah. So this is what nucleic acid. Nucleic acids will be of two types. One of them is known as what? There is a DNA. Okay. That's the full form is deoxyribonucleic acid. And RNA is known as a ribonucleic acid. Perfect. Nucleic acid is made up of what? It is made up of polynucleotide chain. Polynucleotide chain. I'm telling you very from the very first beginning of this chapter, I'm telling you, there are so many people, not only the students, but even some teachers are also making mistake. I mean that the biology teachers are making mistake. That is what they are, in spite of saying polynucleotide chain, by slip of tongue, they are saying polypeptide chain. Polypeptide chain is also a right word which will give rise to a protein. But here we are going to synthesize RNA and DNA. Okay, yeah, so this will be having a polynucleotide chain, not polypeptide chain. Okay, so again, nucleic acid is what? Deoxyribonucleic acid. Another one is a ribonucleic acid. So DNA and RNA. And I told you that don't forget to say polynucleotide chain, because polypeptide chains are present in the protein. So the whole chapter is about the DNA and RNA. But in the end of the chapter, there will be a translation in which polypeptide chain is being formed. Polypeptide chain is being formed. Okay, so in the translation polypeptide chain is being formed. Okay, but here it is polynucleotide chain. Okay, what do you mean by poly? Many. So many nucleotides will combinedly give rise to polynucleotide chain. Okay, many nucleotides give rise to polynucleotide chain. Yes, so poly means many. When so many nucleotides will combine with each other, then they will give rise to a polynucleotide chain. Okay, so in the case of DNA, there will be two polynucleotide chains. Can you see? In DNA, there are two polynucleotide chains. Okay, so one is going like this. So one is going like this. So this is one polynucleotide chain. and other going like this. Yes. So in the DNA, there are two polynucleotide chains, while in the case of RNA, there is only one polynucleotide chain. Okay. Poly means many. Many nucleotides will give rise to this chain. So what do you mean by one nucleotide? Nucleotide is made up of a pentose sugar. What do you mean by pentose sugar? The sugar which is putting on pent. हैं जो पेंट पहन के घूमती है वो नहीं बाबा पेंटोस बोले तो 5 कार्बन containing okay as it is containing 5 carbons that's why it is known as a pentose sugar okay so it will be having how many carbons babas and babies 5 carbons so as it is having the 5 carbons that's why it is known as what it is known as a pentose sugar. Now you may ask me that where are the 5 carbons? Okay, all corners are carbon. 1, 2, array 2, 3, RAFO, and 5. Hmm. Gone. Outside gone. So okay. So you may people wondering that sir, what's going on? So one small cutie Stuart Little was over here, okay, and just gone out. Okay, so this is what 1, 2, 3, 4, 5. So 5 carbons are there, and that's why this is known as what? This is known as a pentose sugar. If I'm writing over here D for Delhi, then it is not D for Delhi, but D stands for deoxyribose sugar. And if I put R, then it is R for ribose sugar. Perfect. Okay. Yes. So pentose sugar could be of two types: ribose, which is present in the RNA, and just remove one oxygen from ribose, then it will become deoxyribose. That is known as a DNF. Perfect. Yes. Okay. Now, nitrogen base will be of two types: purines and pyrimidines. What is the difference between purine and pyrimidines? I'm going to tell you. Okay. But whenever the tide is written, don't forget to add phosphorus. So if the tide is written, that is because the phosphorus is attached to it. If phosphorus is attached to it, then only it is known as a tide. And if phosphorus is not there, then it is known as a chalvakaside. Eh? Then it is known as a nucleoside. So I believe it, I mean that in the case of nucleotide, nitrogen base, pentose sugar, as well as phosphorus is present. But if it is nucleoside, then phosphorus is not there. Oh, so phosphorus is not there, then what is there? So it's having a pentose sugar and nitrogen base. That's it. Okay, so that's known as a nucleoside. Perfect. Agree? Okay. Now I will give you some much more detail about the nitrogen base also. So now I'm just looking on the nucleoside. So nucleoside is having the pentose sugar. So I already told you about the pentose sugar. Which are those pentose sugar? That is ribose and deoxyribose. Okay, and nitrogen base. And there is no phosphorus. If there is no phosphorus, then only it is known as a nucleoside. Perfect. Yeah. Okay. So nitrogen base will be of two types: purine and pyrimidine. Purine will be made up of a two-ring structure. Purine is made up of what? Two-ring structure. Pyrimidine is made up of what? Single ring structure. So if there are two rings, then it is known as a pyrimidine. Sorry, it is known as a purine. And if it is only single ring, then it is known as a pyrimidine. Perfect. What are the examples of the two-ring structure? That is adenine and guanine. And it is denoted by this kind of an arrow. Okay. And pyrimidine is a single ring structure which is example is a cytosine, thymine, and uracil. Cytosine, thymine, and uracil. Cytosine is common in all organisms, all the nucleic acids. Okay, so cytosine, adenine, and guanine, these three are common. Okay, they are present in DNA also, and they are present in RNA also. Okay, but in the case of DNA, thymine will be present. In the case of RNA, uracil is present. Again, in the case of thymine, thymine is present in the case of DNA, and uracil will be present in the case of RNA. Okay, so how can we keep this in our mind? So as per the English grammar, if you are saying you, then what will be the verb after you? R stands for RNA. So you always bind, U is always present in RNA. So uracil is always present in RNA. So what is remaining? That is thymine. Thymine is present in DNA. Again, I repeat this memory technique. U, then after there will be R. R is what? It's a verb. So uracil is present in RNA. That's it. So U is present in RNA. Perfect. Done. Okay. So U and then after there will be R. Okay. And thymine is present in DNA. Perfect. Yes. So now you can see that this is known as a whole nucleic acid, and this is its formation. Wow, it's really a good one. What do you say? Huh? So let me just put it in a particular way so you can see the whole sequence very nicely and can copy it. So I would just request you all now to copy this sequence, okay? Because this is very fundamental one. Actually, you should know about from your knowledge of the 11th grade, okay? But I also know that you consider that 11th is not important. Yeah, is it visible? Yes. So just pause your video and please write down this. As you complete this, just write down WW. Okay, so I am again repeating this sequence that nucleic acid is a DNA, that is known as a DNA is a deoxyribonucleic acid. RNA is a ribonucleic acid. They are made up of a polynucleotide chain. Poly means many. Many nucleotides combine and give rise to polynucleotide chain. Such a two polynucleotide chains are present in the DNA and one polynucleotide chain present in the RNA. Nucleotide is made up of what? Pentose sugar. Pentose means what? It is having a 5 carbon. There are two varieties of the pentose sugar. One of them is known as a ribose sugar, which is present in the RNA, and there is another one which is known as a deoxyribose that is present in the DNA. Then the nitrogen base, there will be two types of the nitrogen bases. One is a purine, another one is known as a pyrimidine. And if it is written a tied, its meaning is that something is being tied with this structure, and that's what the phosphate. Yes, so phosphate is tied with it, and that's why it is known as a nucleotide. If you put the phosphorus in the side, then it will be known as a nucleoside. So that's what the nucleoside is, having a pentose sugar and nitrogen bases. Nitrogen bases will be having a two ring structure, which is known as a purine, and single ring structure is known as a pyrimidine. The examples of a purine are known as adenine and guanine, and it is denoted by such arrow. And pyrimidine is known as a single ring like structure, and its examples are cytosine, thymine, and uracil. Uracil, U is present in RNA and T is present in DNA. Yo, okay, so just keep this sequence in your mind. This will help you a lot. And now let's study about each and every component slowly and gradually. Yo, okay, so let's study about each and every component over here. Yes. Okay, so that's what it was over here. Just a minute, give me a yes. So you can see that here polynucleotides are polymers of nucleotides. Poly means many. DNA and RNA are polynucleotides. A nucleotide has a three component, as I told you: nitrogenous base, that's of the purine and pyrimidine, pentose sugar, ribose in RNA and deoxyribose in DNA, and phosphate group. Yes or no? Now, the structure of polynucleotide chain. In the structure of polynucleotide chain, you can find out some basic nitrogen bases are present. One of them is a purine. Purine is a two-ring structure, which examples are adenine and guanine, and pyrimidine is a single ring structure, which is known as a cytosine, thymine, and uracil. Am I right? Purine is two-ring structure, that is adenine and guanine. Pyrimidine is cytosine, thymine, and uracil. Yo, everybody is okay with this? Yes or no? Yep. The next is about what? The next is about the structure of polynucleotide chain is a uracil. So, we have to study about the nitrogen base. Uracil is absent in DNA, but it is present in RNA. And I have given you a memory technique also to keep this in your mind. That is what? If you are writing U, then after U, there will be a R only. Okay. And let me write down U in a fashion. U. Then U is connected with R. And I'm writing R also in the fashion. Okay. Yes. So instead of uracil, thymine is present in the case of DNA. So thymine is absent in RNA and present in DNA. Okay. Instead of thymine, the uracil is present. Perfect. Erwin Chargaff's rule is saying in DNA, the proportion of A is equal to T and the proportion of G is equal to C. Why? Because whenever you are free, go to the CG road of Ahmedabad city. So see you at CG road. Eh? At, what do you mean by that? At CG road. So A bind with the T and C bind with the G. A bind with the T with the help of a two weak hydrogen bonds and C bind with the G with three weak hydrogen bonds. So again, I am giving you a memory hook. That is what at CG road. So whenever you are getting a time, just go to the CG road and see you at CG road. It is also a nice place where you can find out a municipal market where my favorite pizza are there. That is known as a Wah pizza, as well as you can enjoy the thick shake of Cheers. Okay. Yeah. And as per the season, you just ask that which one will be the fresh one. That will be the best one. And another option is Choice also. Choice restaurant is also a very good one. And you can enjoy lots of things over there. Okay. And don't forget to go for the shopping. Yep. Okay. So here in the single slide, I am telling you two memory technique. One is UR, that is uracil is combined with the RNA, is present in the RNA. Okay. And at CG road. Okay. So the proportion of A is equal to T and the proportion of G is equal to C, because A always bind with T and C always bind with the G. That's how it is what at CG road. Okay, so R as well as the at CG road, these are the two memory techniques. Okay, yeah. So still there are lots of the slides I am going to show you on the one concept only, whatever I have already explained you. Okay, but yes, I have to show you about all those, and that's why I'm just showing you. So here you can see the nitrogenous base is linked to the pentose sugar through night and glycosidic linkage. What do you mean by that? So nitrogen base plus pentose sugar is known as a nucleoside. And the examples of different nucleoside is adenosine. That is in the case of if R is a ribose sugar. And if the D is as a pentose sugar, then the name will be deoxyadenosine. Are you getting it? Okay, so its meaning is that let me make it over here very clear. Okay, wow. So you can draw very nicely, na? Yes, because I have selected such a pen. That's why such a straight lines are going on. Okay, so here you can see if it is R as a ribose sugar, then these all will be applicable. Okay, if as a pentose sugar we are using ribose. Okay, yeah, and as a pentose sugar if we are using deoxyribose, then the other bracket things will be considered. So this is for ribose, this is for ribose, and this is for deoxyribose. Hey, let me change the color. Suggest me a good color, babas and babies. The blue color is already there. Red I have already used. Brown, okay. So these all will be what? These all will be having the deoxyribose as a pentose sugar. These all will be having what? Deoxyribose as pentose sugar. So in spite of this R, there will be a D. In spite of R, there will be a D. D for Delhi. Yo! Okay. Now, I'm talking about the bond which is known as a glycosidic linkage. So where is it? Okay, so the glycosidic linkage is over here. This black colored one is known as a glycosidic linkage. So this is known as a glycosidic linkage. Yes, are you getting this? This is known as a glycosidic linkage, and it is connected with what? It is connected with the A. A. A means adenine. If adenine named nitrogen base is connected over here, then this is known as a adenosine. If R is a ribose sugar, and if the pentose sugar is a deoxyribose, then it is known as a deoxyadenosine. Are you getting this? Everybody okay with this? Yes. So again, I am just asking you that in between the pentose sugar and nitrogen base, which chemical bond is present? That is known as a N-glycosidic linkage. Okay, N-glycosidic linkage. Perfect. Okay. And nitrogen base and the pentose sugar are connected, and that will give rise to a nucleoside. So this is nucleoside, and if I just want to make this a nucleotide, then I have to attach one phosphorus to it. Okay, I have to attach one phosphorus to it. Okay, so let me show you here the number of the carbon atoms. This is one. What comes after one? Two. This is 30. Okay, it's not visible. Okay, just a minute. This is fourth, and here will be the fifth carbon. Okay, as okay, I'm just using another color. There is one. Is it visible? This is two. There is a three, fourth, and the fifth carbon is over here. Okay, And to this fifth carbon, phosphorus is being added. If phosphorus is added, then it is known as what? Then it is known as a nucleotide. Very good. Okay. So let's go further here and let's give rise to a nucleotide. So we are enough done with the nucleoside. Yes or no? Okay. Yeah. So let's go further and let me change the slide. Yeah. See, this is what the pentose sugar. Okay, 1, 2, 3, 4, 5. Very good. Then to the first carbon, if the nitrogen base is added, uh, this is what N glycosidic bond. Yes, perfect. Very clear. Okay. Then after here, these are the nucleosides: adenosine, thymidine, guanosine. Then this is what the deoxycytidine. Okay, so here will be a D. Okay, or it is a cytidine. So if we are assuming R over here, then the first names are there. If you are assuming the D over here as a pentose sugar, then the brackets name you have to consider. Yes, sir, here there is no bracket name, of course, because the thymine will be present in only in the case of a DNA, and that's why it is not given in a way. Yo, everybody okay with this? So, as you can see that here the arrow is like this because it is a adenine. So most of the purine will be like this. Okay. Now, the nitrogen base plus sugar plus phosphate group. If phosphorus is added, then it is known as a nucleotide. Two nucleotides linked with each other with the help of a phosphodiester bond, and that will give rise to dinucleotide. Now, the question is that, sir, what do you mean by the 3' and 5'? So, first nucleotide's third carbon combined with the next nucleotide's fifth carbon. Again, first nucleotide's third carbon combined with next nucleotide's fifth carbon. That is known as a 3 days, 5 days. More nucleotides are known as a polynucleotide chain. Kallu here. Arey, look here. The phosphorus is over here. Yes, so now you can see that this is known as a nucleotide. So sugar plus nitrogen base plus phosphate. How is it? This bond is known as what? This bond is N glycosidic bond. Okay. Then now you can see these are the examples of the nucleotides. Okay, so here, okay, let me write down over here that these are P. Bisika P. So this is what P. Okay, so that's why it is a nucleotide. Yes. Okay. Now, if you are talking about 3' and what was that? 3 to 5 now. So it's just like this. Look here. This is first carbon. This is second carbon. Second carbon. This is third carbon. This is fourth carbon. And this is fifth carbon. So third carbon of first nucleotide combined with next nucleotide's fifth carbon. So let me label here also the carbon numbers. 1, here is 2, here is 3, here is 4, and here is 5. So now you got it. First nucleotide's third carbon combined with next nucleotide's fifth carbon. Again, first nucleotide's third carbon combined with Next nucleotides, fifth carbon, and this bond is known as a phosphodiester bond. This bond is known as what? Phospho. बोलो नहीं यार. Again. Phosphodiester bond. Phosphodiester bond. So it is phosphodiester bond. Yes. Now you got it. What do you mean by the 3' and 5'? Everybody okay about this? Krina, Shreya, you both are okay about this? Say yes if you are.
Yes, sir.
Okay. Thank you. So let's move further. Here. So this is about the things which is about the DNA. Okay. So Frederick Miescher, as a 1869 identified DNA and named it as a nuclein. Why it is given such a name nuclein? Because it is found in the nucleic acid nucleus. That's why it is known as a nuclein. Okay. James Watson, Francis Crick proposed a double helix model of DNA. It's a polymer of deoxyribonucleotides. So there are two polynucleotide chain. One is going over here. Just look here, babas and babies. Look here. There are two polynucleotide chains are going on. Okay. One polynucleotide chain is this one. Just look over here. This one. Yes. Okay. So one polynucleotide chain is going in this way. Yes. And the other polynucleotide chain is going in this way. Can you get this? It's just like a famous hairstyle of the girls. That's the chotla. But in that there will be 3 flicks. Here there are only 2 flicks. Mind well. Okay. So let's go further now. Oh. Length of the DNA is based on the number of nucleotides present in it. Okay. Yeah. Length of the DNA is based on the number of nucleotides present in it. That is phi 174. It is a type of a bacteriophage. What do you mean by bacteriophage? What is it? Is it a bacteria? What is it? Huh? Anyone can speak out. Yes, what is bacteriophage? So this is how your relevance is very bad, and it's not about only 1 or 2 people. There are lots of the people who are not answering this. Anyone? It's a structure of virus. Very good. Which virus are known as a bacteriophages? Okay, so the virus is whose host cell is bacteria is known as bacteriophage. Got it? Okay. In the 5174, a bacteriophage, it is having 5,386 nucleotides. Oh, then there are so many different types of the bacteriophages. Like one of them is known as a bacteriophage lambda, and lambda is having a 48,502 base pairs. Base pairs, what do you get by base pairs? A and T. So that is known as a one base pair. Are you getting it? C and G, that is another base pair. So that's how the base pairs. Okay. So bacteriophage lambda is having a 48,502 base pairs. E. coli bacteria, hey, do you know about the E. coli bacteria? Yes, it's known as the Escherichia coli bacteria, has 4.6 into 10 raised to 6 base pair. Oh, so many base pairs. Okay, haploid content of a human DNA. Haploid content means what? Single N. Okay, then it is 3.3 into 10 raised to 9 base pair, and if you will go for its double, that is known as a 2N, then it will be 6.6 into 10 raised to 9 base pair. Okay, this is very important. Just keep this in your mind. Okay. Are okay? Yeah, and there are so many MCQs are being asked, so many questions are being asked just from this. Okay, so don't ignore this paragraph, it's a very important one. Here, the next is about what? So it's about the detailing of that DNA only. Okay, so it is made up of a 2 polynucleotide chains. Already we have just completed it, so I'm just showing you where the backbone is made up of a sugar and phosphate, and the bases are projecting inside. So its meaning is that, just think about the staircase. It's having supports. So this is one support, this is another support. Okay, those supports are made up of what? Made up of a sugar and phosphate. Okay, and what is present in between them? So I'm just putting my digital pen in between them, and this is what? This is known as a nitrogen bases. This is known as what? Nitrogen bases. So these are nitrogen bases. So you just think about this, so many things are like that. So it will give rise to a ladder. Yes or no? Are you getting it? It's just like a ladder. Okay, yeah. And let me show you. So there is a one ladder. Okay, and let me take one ladder in my hand. There is another ladder in my hand. I am just holding it like this, and now twist it. When you will twist it, it will give rise to a double helix structure, as like a spiral ladder. So many times you people are also making the DNA in the decoration, but you never know about that. In the birthday decoration, what we are taking that the one different colors, and we are putting a tap on one end, then we twist on the other end, and then after we put the tap on the other hand, and that's what we are making the DNA. Are you agree with me? Yes, no? Okay, so especially Shreya and Krina, are you agree with me, beta?
Yes, sir.
Okay. So one chain will move like this, and another chain will be reverse of it. Another chain will be reverse of it. Okay, and that's why one chain is known as a 3' to 5', another chain is known as a 5' to 3'. Okay, let me show you how and what I want to convey you by 3' to 5' and 5' to 3'. Okay, yeah. So let me show you one of the another structure, so you will get to know about it. Okay, let me check if I can show you. Otherwise, I have to draw it, and that will take a time. Okay, can you see this? Yes, so here, just changing. One is what? 3' to 5'. Another one is a 5' to 3'. But why is it so? Why it's inversely proportional? So, that's what I'm going to show you. That why it is known as an anti-parallel chain. Okay. Yeah. What do you mean by anti-parallel? Okay. So let me show you by some other photographs, whatever I am having. Okay, just stay with me. Okay, so let me show you the what do you mean by the 5 days to 3 days and 3 days to 5 days. Okay, so yes, see here you can see that the backbone is made up of what? The backbone is made up of just only phosphorus and sugar. So yes, this is known as a sugar and phosphorus, sugar and phosphorus, sugar and phosphorus, sugar and phosphorus. Yes or no? In between here, you can find out the steps are there. Those are made up of a nitrogen base pairs. Okay. And here it's like you can see that this is what first nucleotides, which carbon is joining. So, this is carbon number 1 to which the nitrogen base is attached, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. So, this is fifth carbon combining with third carbon of the next nucleotide. Fifth carbon of first nucleotide combined with the next nucleotide's third carbon. Okay, so that is known as the 5 days to 3 days. Again, this upper one is having a fifth carbon and lower one is his third carbon. So that's why this is known as what? This is known as a 5 days to 3 days. So you can see always it is just like 5 prime to 3 prime structure. Perfect. Everybody okay with this? Are you agree with this? Let me draw a very good line so that you can see that where the sugar and phosphate are also present. Just a minute, beta. Okay. So this is what sugar and phosphate backbone. Yes. On this side also, I can tell you about sugar and phosphate backbone. Are you agree with me? Yes. And that is just acting as a supports in the ladder. Okay. And in between these are the steps. And the moment you are taking both the supports in your hand and just twist it so that it will give rise to a DNA. So which is a helical structure, which is known as what? Helical structure. I told you A bind with T by 2 weak hydrogen bonds, C bind with G with the help of a 3 weak hydrogen bonds. So here we can see, yes, look over here, that is carbon number 3. Can you see? See, this is 1, 2, 3. Okay, so that's where here and here. So it's a mirror image. So it's a 1 because to the 1 carbon only nitrogen base is attached. Are you getting it? So this is carbon number 1. Are you getting it? This is carbon number 2. This is carbon number 3. This is carbon number 4. And the carbon number 5 is over here. Are you getting it? So here you can see the first carbon attached with the nitrogen base and fifth carbon attached with the phosphorus. So here you can see that first nucleotide's third carbon combined with next nucleotide's fifth carbon. Again, first nucleotide's third carbon combined with next nucleotide's fifth carbon. Yes or no? Are you getting it now? Everybody? No, Tirali is not okay with this. Thanks for being so much transparent, beta. Okay. I repeat again. Here, what we are talking about, there is one word which is known as an anti-parallel. I want to explain this word. What is that? Anti-parallel. Okay, what do you mean by parallel? We already know that the parallel lines are the such a lines which will not intersect each other. They will never intersect each other. So if they are not intersecting each other, then it is known as what? Then it is known as a parallel. But here the word is anti-parallel. What do you mean by anti-parallel? So they are moving in the opposite direction. Okay. So as I mentioned you earlier in the same chapter, that to the first carbon of the pentose sugar, nitrogen base is attached. Yes or no? Okay. So whenever you will find out the nitrogen base is attached, that will be given a number 1. Okay. Yes or no? So let me give this number 1 for this. This is number 1 carbon for this. For all nucleotide, I am giving you the numbers. Hello. Then here you can see this is carbon number 2. Yes, is it 2? Are you agree with me? Keep speaking. Tirali agree. This is number 3. Okay, so here it is 3. Okay, here it is 3. Here it is 3. Okay, then afterward, this is known as a 4th one. This corner is 4th. And this is 5th. Iha. Again, this is what? 4th. And this is known as a 5th. Again, this is 4th. This is 5th. This is 4th. This is what? 5th. Are you agree, beta? Let me put such numbers over here that will be helpful to you in this way. Okay? Yeah. So to whomever the carbon, the sugar is attached to the nitrogen base, that is carbon number 1, and the sugar is attached to the phosphorus, that is carbon number 5. Yes. Okay. Okay. Let me write it down, and be with me. Be transparent. Okay. This is where the nitrogen base is attached. So this is carbon number 1. This is 1 for everyone. 1, 1. Yes or no? This is carbon number 2. This is 2. This is 2. This is 2. Then this is third one. Are you getting this? This is third one. This third one. Then this is fourth one. This corner is fourth one. This corner is a fourth one. This corner is a fourth one. This corner is a fourth one. And what you can find out in between, that is the fifth one. So this is 5. So, sometimes it becomes very difficult for me to write down 5. That's why I'm just writing F. I hope it will be easy for me. Even that is also difficult. Yes, so this is known as a fifth one. Okay, so this is known as a fifth one. And here you can find out the fifth one. Perfect? Yes. Now, first nucleotide's third carbon, first nucleotide's third carbon combined with next nucleotide's fifth carbon. Can you get it? First nucleotide's third carbon, that is this one, is combining with next nucleotide's fifth carbon. Again, look here. This nucleotide's third carbon combined with next nucleotide's fifth carbon. That is this one. Yes or no? First nucleotide's third carbon combined with next nucleotide's fifth carbon. Yes or no? Sure. That's why this is known as first nucleotide's third carbon. That's why it is known as a 3 prime. Okay. So now she is telling me that she is confused in between 3 days and 5 days. Okay, no problem, no problem. You got about this line, this right hand side is perfect. Now let's look on the left hand side. This is carbon number 1, 2, 3, 4, 5. First nucleotide's fifth carbon combined with next nucleotide's third carbon. This is first nucleotide's fifth carbon combined with next nucleotide's third carbon. Again, first nucleotide's fifth carbon combined with next nucleotide's third carbon. Are you getting it? That's why this is known as a 5 prime to 3 prime, and this is known as a 3 prime to 5 prime. So this is how it is known as an anti-parallel. So they are parallel, but moving in the opposite direction. So it's just like this. Let me show you simply. I can show you like this. This is what they are parallel, but they are moving in the opposite direction. Are you getting it? As they are moving in the opposite direction, that's why it is known as an anti-parallel. Yeah. How is it? Perfect. Are you getting it? Thanks for asking this question. I really appreciate. Okay. Yeah. So feel free to ask me any kind of a question whenever you are not getting anything. Don't move your head like this. Be frank and just let me know that you are having a problem over here. So this is known as a 5 dash to 3 dash, and another side is having the 3 dash to 5 dash. Perfect. How is this? Perfect, ne? Bolo. Everybody. Okay. So, that's why I'm just telling that what kind of things are given in the book, and I just want to show you and want to teach you the exact meaning of it. At least we have to complete in this particular lecture at least about the DNA. Yes or no? At least we have to complete about the DNA. Babas and babies, are you agree with me? Yes. Okay. So let me read again and let me tell you about what the theory is telling. So we have completed up to what? Yes, up to here. The Chargaff. Yes, and the different dimensions. Okay. So now we are going over here and telling you it is made up of a 2 polynucleotide chains where the backbone is formed of the sugar and phosphate and the base is projected inside. Yes. Okay. They are making the steps of the ladder. Yeah. Okay. Then the two chains have an anti-parallel polarity. So now we got it. What do you mean by anti-parallel? So anti-parallel means what? Parallel means they are not intersecting with each other. That is clear. Okay. And one is moving 5 prime to 3 prime. So first nucleotide's fifth carbon combined with next nucleotide's third carbon via phosphodiester bond. And in the opposite side, first nucleotide's third carbon combined with next nucleotide's fifth carbon. So one is moving 5 prime to 3 prime. Another one is moving 3 prime to 5 prime. Agree? The bases in the two strands are paired with the hydrogen bonds forming base pairs, which is known as a at CG road. So A bind with T with the two hydrogen bonds and C bind with the G with the help of a 3 hydrogen bonds. Purine comes opposite to the pyrimidine. That's why A bind with the T. A is adenine. Adenine is a purine, and T is a thymine, which is pyrimidine. This gives a uniform distance between the two strands. The two chains are coiled in a right-handed fashion. So that's why it will give rise to what? So, I told you that there are two ladders. Okay, it's a ladder, single ladder, and you have to just twist it. The moment you will twist it, it will give rise to a coiled structure. So it will be just like a spiral ladder. Yes or no? The pitch of the helix will be 3.4 nanometer, or is it 34 angstrom. What do you mean by pitch? I will tell you. Number of base pairs in each turn will be 10, and that's why the distance between the two base pair will be 3.4 angstrom. See, in one pitch there are 10 base pairs. Okay, and the distance of one pitch is 34 angstrom. So the distance between two consecutive base pairs will be what? So 34 by 10, that will be 3.4 angstrom. Yes or no? Okay. So here you can see this is known as a one pitch. The pitch is starting from here to here. Okay, again I am telling you from yeah, let me take from here to here. So this is known as what? This is known as a one pitch. Eh? This is known as a one pitch, and that is what 34 angstrom. So if this is 34 angstrom, its meaning is that the distance between two consecutive pairs will be definitely 3.4 angstrom. And you know how to convert the angstrom in the nanometer and all those, thanks to physics subject. Yes or no? Yes, so here the distance will be what? 3.4 angstrom. Yes, so this is known as what? 3.4 angstrom. Yes, so this is what it is. Are you getting it? Yeah, everybody okay with this? So this is what the sugar phosphate backbone, and these are the base pairs which is acting as the steps. None? Okay. Agree with this now? Okay. So this is just I just shown you that how one pitch is created, and in such a way the whole base pairs will keep going on and will give rise to a whole DNA. Okay, so this is what the DNA, and A to T, C to G, and that's what that's going on. So you can see again, I am just showing you that's 5 prime to 3 prime. Can you see now? 5 prime, this is 5, this is 3, this is 5, this is 3, 5 prime to 3 prime. And here is a 3 to 5. Here it is 3 to 5. Here it is 3 to 5. Yes or no? So here it is 3 prime to 5 prime. Here it is 5 prime to 3 prime. So that's how it is anti-parallel to each other. Hai bhai? Are anti-parallel to each other. Again, this is known as the first nucleotide's third carbon combined with next nucleotide's fifth carbon. Okay. Say okay. If you are getting the, then only say okay. Are you getting it? This is what fifth carbon combined with next nucleotides, third carbon. This is the third one, Baba. This is the fifth one, Baba. Are yaar. Should I repeat this? No or yes? No. Okay. Otherwise, I have to speak the same throughout the night in my sleep. And perhaps you will also speak the same thing in your night. Okay, yeah, so let's go further, and this is what about the DNA. Yo, so here again I'm just looking forward. Here you can see this is a very good this is, you can see the first, second, third. Okay, so third nucleotide combined with next nucleotide fifth carbon. First nucleotide third carbon combined with next nucleotide's fifth carbon. Yes or no? Babas, baby. These two combine with each other. You just look anywhere you will find out at CG road, A to T. C to G. Yes or no? And vice versa. And vice versa. So A will bind with T, T will bind with A, C will bind with G, G will bind with C. Yes. Wow. In between A and T, there will be two hydrogen bonds, two weak hydrogen bonds, and in between C and G, there will be three weak hydrogen bonds. Okay, and this is what I already explained you. Okay, now the length of human base pair. But okay, I think for the starting of this chapter, it is enough. And I would like to stop over here because this was just only a revision of whatever you know from your 11th grade. There is no any new thing I have taught you. But yeah, fundamental clarity you got by today only. You know, in the 10th also the words were given that DNA is anti-parallel and so on. But yeah, you got what you mean by that. Okay, so just a minute. Okay, so I hope you enjoyed a lot and I also enjoyed a lot. Yeah, bye bye.